The teaching of Vedantu was amazing. One aspect where Vedantu teaching differed from a normal offline coaching was the attention teachers gave to our doubts. No matter how many times we asked a doubt, the teachers would patiently clear the doubt every single time. And they made learning a very fantastic experience for us.
Hello my dear children, Namaste and welcome to yet another biology session of the Class 10 ICSE series. How are you all? Hi SD classes, hi Aditya, hi Ahana, hi Hiral. I can see a lot of familiar names. I remember all of your names. I'm quite good at remembering things, okay? So I notice all your names. Thank you very much for joining me today, guys. So as you can see, oh well, before that, let me introduce myself once again. I'm Ambika, Master Teacher of Biology right here at Vedantu. How many of you attended my session, my previous session today, the motivation session in which we talked about procrastination? Yes, good evening, top 10. Yes, Ahana, I do remember you. Yes, of course, I remember a lot of your names. You did attend it, is it? Ram Sevak? Okay. Those of you who haven't, please do go back to the video and make sure you watch it. I'm sure you will get some takeaway from it. We talked about how precious time is and about the general tendency of people and the general tendency of students to procrastinate and not have motivation to just get started. Okay, so that's all right. Top 10, you can have a look at that. When you have some time. But anyway, today continuing with our regular biology sessions, our biology syllabus, you know that we are done with the first chapter, cell cycle and cell division, and the second one, which was structure of chromosomes, which you can consider as another chapter or as a second part of the same chapter. We have seen how cell cycle happens, how mitosis happens, how meiosis happens. Hi all of you. Thank you very much for joining me. We are just getting started, so let's hope more people will join us in the next few minutes. Anyway, mitosis, meiosis, and we have seen what the structure of chromosomes is, what genes are, what DNA is, what chromatin is, all of these. Okay, and remember, I also told you that it is very important to get all these terms clear in your head before you attend my session in the genetics topics, because we will be using the terms homologous, chromosomes, all of these things interchangeably. We may not have the time to just sit and revise all the concepts over and over again. Just in case you have missed any of those sessions, it doesn't matter. For now, you can stay with me for the next 40-45 minutes. I'll make sure you understand all of this, but in case you need a little more help about the basics which we did last class, in the previous classes, please do make it a point. Please be sure to go back and have a look at those videos once again, because again, these basics are extremely important to understand, and just mugging up doesn't help. Okay, so in case you are the kind of a student who thinks that biology needs only mugging up, I'm telling you that's not the case. There are a lot of basics. If you understand right, the rest of the subject will be very, very interesting and very, very easy for you to remember. Okay, so our ultimate aim is to score the best for your board exams, and we have already started the preparations for it. Let's continue. Okay, but before that, I have something to tell you. The only person you are destined to become is the person you decide to be. That is amazing, right? Which means that the control is totally in your hands. Whoever you decide to be, that is who you are destined to be. So decide right now, children. Like I told you in my last motivation session, do not waste any more seconds. Do not waste a minute at all of your time. Do get started and decide who you want to be. That is what you will be ultimately. I'm sure you will be that if you get started right. Okay, and now coming to the homework question that I gave you in the last class. Yes, thank you very much, all of you who have joined me right now. Even if you are from CBSE, it doesn't matter, because CBSE children, you also have a topic on heredity, right? Heredity and evolution. So I'm sure this will benefit you also. Okay, so stay with me, CBSE children also, and state board, if any of you are from state boards, in case this is in your respective state board syllabus, heredity or Mendel's experiments, please do stay with me for the next 40-45 minutes. I'm sure you will also benefit from this. Okay, let's just get the basics right. The more you hear about it, the better you retain it in your head, right? Let's look at this question once again. Yes, thank you very much, Shivam. If the DNA sequence on one strand read ACGTTCGAATC, the sequence on the complementary strand would be. And yes, I did get a lot of answers which were completely correct. I did not want to just put one or two names this time. Some of you have been telling me that you also gave me the correct answer, but I did not put your name here. It's for the simple reason that I don't want to overcrowd the slide with a lot of names. I have been seeing all your comments, and all of you who have been answering my homework questions know for sure that I have been replying personally to each of you, and some of you also mail me personally, right? And all of you know that I also reply to all the emails that I receive, maybe not immediately, but whenever I find some time. So this time anyway, I thought of putting most of the names of the children who have given me the correct answer. Okay, so as you can see, the answer is TGCAAGCTTAG. And okay, before we look at the names, let us see if the answer is correct. How many of you remember this, and how many of you understood complementary base pairing in DNA? I hope all of you have understood that. Okay, yes, so tell me. Just remember the simple rule: A pairs with T and G pairs with C, considering that the complementary strand of the given sequence, this given sequence, will be A to T, C will be G, C, A, G, C, T, T, A, G. Yes? Okay. Once again, let us look at the names. We have correct answers from Shashi. We have the answer from Siddhi Sharma. I have brought your name before also, Siddhi. I remember you very well. Ayushi again, and Isha, Karanbir, Ahana. Thank you very much, guys, for giving me the correct answer. Here, right now, we don't have the time to take up that topic and explain. Just in case you have missed that session, please do make it a point to watch the video on the topic structure of DNA, which was my session on Friday last week. Okay, and even after that, if you have any questions, post in the comment section or send me an email. I will definitely explain it again to you. Okay, so let us go ahead with what we have to discuss today. Before that, let's have a quick quiz. How many chromosomes are found in a typical diploid human cell? 23, 46, 22, 44. Yes, 46. Correct answer. Gajanan, 46. 23, 22. Okay. I'm talking about a typical diploid human cell. Diploid means very clearly that it has two sets that a haploid cell would have, and the answer is definitely 46. Okay? Yes. Thank you, guys. Let's look at the next question. Which of these is true about homologous chromosomes? Attached by centromeres, separate during meiosis 1, separate during mitosis, 23 pairs in human males. Okay, let's see who gives the correct answer for this. It's slightly harder than the first question. Okay, A, D, 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 D. Okay, I'm surprised I'm getting D as the answer mostly. I saw one answer that's B. Okay, that really shows that you either have not understood anything about meiosis, or you have not attended my session about meiosis. I can just see two people who have given me the answer as B. 3. Now, Parthiv, Mansi, and I saw a name that scrolled up. I'm very sorry I missed your name. But yes, B is the correct answer. Homologous chromosomes separate during meiosis 1. What is attached by centromere? It's the sister chromatids. And what separates during mitosis? Again, it's sister chromatids. 23 pairs in human males. Why is that not the answer? I'm asking you about homologous chromosomes. Human males have 22 pairs of autosomes which are homologous, while the sex chromosomes are X and Y, which are not homologous. Whereas in a human female, it would be 23 pairs of homologous chromosomes. Okay, so right here, among all the options that we have, the correct answer is B. The next question is not based on something that we have discussed in any of our sessions, but hopefully something that you know from your lower grades. Let's see how much of that you remember. Which of these is the sexually reproductive part of a plant? Leaf, flower, stem, and root. Flower, yes. Niku, Kaluri, Pavan, Sunita, Valarmati, yes. B, absolutely B. Yes, the flower is definitely the sexually reproductive part of a plant. Thank you for remembering that. The pollination of a flower by the pollen of the same flower is self-pollination or cross-pollination. What would that be? That's a very simple one. I'm sure you would remember that. Yes, self. A is the answer. Trijal, no, it's not B. The answer is in fact there in the question itself, Suresh Kumar. It's not B. It's pollination of a flower by the pollen of the same flower. So self or the same flower is pollinating it, which is why the answer is self-pollination. Now, why did I give you questions about flower and self-pollination? Because these will be relevant to our session today and you will know as we go about it. So let's get started with our next chapter, which is some basics of genetics. And the topic that we have today is about Mendel's experiments. Hi Vaishali. You are just in time. Hi Saurabh. Okay, now first of all, have a look at this family picture. Those of you who have seen this picture in one of my previous sessions from meiosis, I think, would remember it, would recall this. It's the Simpsons family. If you are into the habit of watching cartoons, if you know cartoon characters, this is the Simpsons family. What is it that you observe about them? Just by one look, you know that they belong to one single family. How is it that they look so similar? Father, mother, children, they have a lot of resemblance with each other. They do have a lot of unique characters also. So what do you think about it? Isn't it amazing? Some of you, again, if you have a brother or a sister, some of you may look exactly like your sibling. Some of you may look very, very different from your sibling, right? I'm sure all of you will agree with me on that. You may even have friends who are identical twins. They may look like carbon copies of each other. Some of you may not be able to identify who's who, but people who are very close to them can perhaps identify. Why is it that identical twins look exactly like mirror images of each other? And why is it that children born to the same set of parents who are not twins but just siblings, they have differences? Some may look like their parents, some may look like the grandparents, some may look like their brothers, some may look completely different. All of those things. Why is it that a cat gives birth to a cat? A human being gives birth only to a human baby. All of these things, right? Even in plants, you see a lot of things. In every generation, it's the same plant that it's giving birth to. You don't really say giving birth to, but it's the same type of plant you would see with every subsequent generation, right? All of this, okay, all of this fascinating science is basically what genetics explains. It's basically the branch of science that explains the process of heredity and variation, basically inheritance of characters from parents to offspring. Yes? Okay, so we'll talk about the laws in the next session. Let's just focus a little less on the chat for now. It is for CBSE also, Ashwin, for your information. This is basically based on the ICSE syllabus, but like I said, CBSE children also have a chapter on heredity, so this will certainly be useful for you also. Okay, so let's go ahead. Please don't feel bad if I'm not calling out your names. I am reading each and every one of your names. If not now, later when I look at the session, I will be reviewing all your chats, all your comments and everything. Umesh, yes, I have seen your comment. Let's focus a little more on the session now. We need to understand a lot of things. Later on, if you have not understood something after explanation, do feel free to ask, but it shouldn't be like you did not pay attention and as a result you did not understand. Okay, so stay with me. Let's look at this. Let's go back to the history of genetic principles. Where did the studies on genetics begin? When did it begin? Well, it in fact began a very long time ago, somewhere in the mid 1800s, in a monastery in Austria. Okay, somewhere in Vienna, where there was a monastery and there was a monk by the name Gregor Johann Mendel. Now, he was a monk, but at the same time, he was also a mathematician and he was very, very passionate about gardening. Okay, so this is Gregor Mendel that you can see here, Gregor Johann Mendel. So, and the picture that you can see here, it's a painting, of course, as you can see. It's been painted in his garden because he was a very passionate gardener and he had a huge, he was growing a lot of plants and he considered his plants as his own children. He was that fond of them. Okay, so Mendel was an Austrian monk and scientist who first worked on genetic experiments, and this was between the years 1856 to 1865, and we call Mendel today as the father of genetics. Unfortunately, Mendel did not get the recognition that he deserved when he was still alive. It was after his death, many years after his death, that science progressed a lot and people really, understood that Mendel had done a lot of intelligent and logical reasoning and had come up with the laws that we know as the laws of inheritance today. Okay, so let us anyway give him a lot of credit. Mendel will be the hero for our session today. Okay, so look at this. These are some terms for the next few minutes, children. For the next 2-3 minutes, please bear with me because we will be learning some terminology, but pay attention because they are very, very important. Every time I mention each of these terms, I will not be able to explain the definition to you. So just pay attention. The first and the basic term that you have to know here is gene. Gene is the basic unit of inheritance located on a chromosome. As you know from our previous session, okay, we have seen what DNA is, we have seen what chromosomes are, we have seen what genes are. Genes are basically segments of DNA. Chromosomes are the carriers of genes. It is in a chromosome, you would see that there are numerous segments of DNA which you can mark as gene 1, gene 2, gene 3, 4, 5 and so on. Okay, genes are basically, if you magnify this, you would see that it's basically a structure, a strand of DNA, and each segment, specific lengths or specific segments of DNA are what you call as genes. Okay, what do they do? Each of our genes carries the information necessary to, express each of our characters. Genes make us what we are. For example, I would have a gene for my eye color. Secondly, I may have gene 2 which codes information for my hair color. Third one for my skin color and so on. Most of these you get from your parents, which is why we see a lot of resemblance between parents and children, right? Okay, next. The next term that you have to know is allele. Allele or allelomorph. Okay, allele is in fact the short form, but allele is how we commonly call it. They are alternative forms of a gene. What do I mean by that? Now the picture that I have here is of a pair of homologous chromosomes. Okay, if you do not recall what homologous chromosomes are, like I said, do go back to the respective session and understand what it is. For now, just understand that in a homologous pair, you get one member from your dad and the other member from your mom. Okay, and these pair together during meiosis, and the rest of gamete formation and everything happens. So, alleles are basically the different forms of the same gene. For example, if I'm talking about the character eye color, okay, I may have in my homologous pair of chromosomes, say for instance, it is gene number 8 that is coding for my eye color, that is deciding what my eye color will be. From my dad, I have got the version of gene which is for black eye color. From my mom, I have got the version of a gene which codes for brown eye color, okay, which is why I have two different letters. In fact, the same letter, but an uppercase and a lowercase letter, indicating that they are just different versions of the same gene, okay. Both of these are alphabet A, just that it's uppercase and lowercase. Okay, why we choose uppercase and lowercase, you will understand as we progress through today's session. For now, just understand that this is my allele for black eye color, this is my allele for brown eye color. This is just an example. It can be any character that you choose. So what is my character? I'm talking about eye color. The trait, the versions of a character are what I call traits, which are black eye color, brown eye color, blue eye color, and so on. Okay, so genes, alleles. I hope that's very, very clear. Okay, let's go on to see what the next term is. It's phenotype and genotype. Phenotype is basically the physical or external appearance of an organism. Like you see in this particular example, the mouse is brown in color, right? So I'm saying it's a brown mouse. Brown is the phenotype that I'm talking about. But what is genotype? Genotype is something that I talk about at the genetic level. Say for instance, if brown is the color, skin color of this mouse, the genotype is what I have represented here in the homologous pair of chromosomes. Maybe because it's brown, I will put it as capital B and capital B, or it can be capital B, small b, whatever it is. But genotype, I'm talking about a combination of the alleles. It is at the genetic level. The genotype here is capital B, and the phenotype is brown. Okay, I hope this is very clear. It's a very important concept. Phenotype, genotype, gene, allele. That is what we have done until now. The next one is monohybrid cross and dihybrid cross. Now, monohybrid cross, like the name tells you, okay, mono indicates that it's one, and di indicates that it is 2. Monohybrid cross is the type of cross in which one single character is studied at a time. Okay, for instance, one single pair of contrasting characters. For example, in this particular example, the character that I have chosen is flower color. Because I'm studying only flower color of that particular plant at a time, I call that a monohybrid cross. Contrasting traits can be seen of the same character. The flower that I am studying can have either purple flowers. The plant that I am studying may either have purple flowers, or it can have white flowers, which are different traits of the same character, flower color. Okay, what about a dihybrid cross? When I study two different characters at a time, I call that a dihybrid cross. Say for instance, in this case, it is seed color and seed shape. I'm considering those two together in my single dihybrid set of experiments. Okay, so I am talking about seed color, which can either be yellow or green. Seeds can either be yellow colored or green colored. Or when I'm talking about seed shape, they can either be round in shape or they could be wrinkled in shape. This is yellow round and this is green wrinkled. It could even be yellow wrinkles, green round, and so on. Doesn't matter. We are trying to define a dihybrid cross wherein two characters are studied at a time. Okay? That's about it. And then coming to the next term, which is homozygous and heterozygous. Now, this is, to understand this, you have to know what homologous chromosomes are and what alleles are. Okay, so these are homologous pairs of chromosomes that I have represented here. And here, if it's a homozygous cross, like the name tells you, homo is similar and hetero will be different. When it is similar, similar what? The similar alleles. The two alleles of the same character are the same, capital B, or even a small b is also the case of a homozygous genotype. Whereas in this case, because the two alleles are different, a capital B and a small b, I call that heterozygous. Homozygous condition and heterozygous condition. Okay? Vaishali, do post your doubt here. If it's relevant to this session and closely related to this, we can explain it. Otherwise, I will explain it once the session is done. Okay? The next one is variation. Now, we did see in the Simpsons family picture also that they did have some differences, right? Like you can see here, it's a rabbit family. All of them are rabbits, but all of them have different colors and different appearances, right? That is what we call variation, differences in the characters among individuals of the same species. But why are they important? They lead to evolution. Imagine if variations did not exist. Human beings on this planet, all of us would look exactly the same. Yes, Shivam, yes, it is possible to study three contrasting pairs at a time. It's called a trihybrid cross, which comes at a much higher level. We'll not be taking up that right now. Okay, so variation leads to evolution because as many generations come up, differences and changes keep accumulating with generation, and over 100 years, over a period of 100 years, you will see that human beings look much different than what we are today. Of course, from our ancestors, our monkey or ape ancestors, we have evolved and become what we are today over thousands and thousands of years. Even in the years to come, well, I can't imagine what humans will look like, but hopefully it will be something very sensible. Okay, so that is the importance of variation. Why is it important? Why does it happen? Basically because it helps organisms adapt to environmental changes and it results in new characteristics, like you can see here. In the case of the giraffe, it is said that giraffes were once short-necked organisms, and when there was a shortage of food on land, on the ground, they had to stretch their necks to reach up to tall trees so that they could eat off the tree branches, the leaves from the tree branches. They kept doing that, and eventually over a period of 100 or 1000 years, they have evolved into the giraffe, the long-necked giraffes that we are familiar with today. Okay, going ahead, we have a quiz time again. Okay, if any of you has been sleeping in my class, it's time to wake up. Let's see who has been actually paying attention. This won't be a very long quiz. The basic physical unit of inheritance is gene, DNA, allele, chromosome. Who's going to tell me the right answer first? Naresh, yes, I've seen your comment. A, B, A, yes, gene. Gene is absolutely the right answer. The basic physical unit of inheritance is gene. The next one, the father of genetics. This you will be able to answer. Mendel, Sutton, Johansen, Darwin. Yes, the answer is very much A. I don't think anyone has a confusion in that. It's not Darwin. Darwin is in fact considered to be the father of evolution, which we will be discussing in later sessions. Mendel is known as the father of genetics. Phenotype. External appearance of an organism, genetic constitution, dominance, recessiveness. What is the answer? A, yes. Any other answers? A, yes, absolutely. The external appearance of an organism is what we call the phenotype. What about genetic constitution? What would you call the genetic constitution of an individual? Yes? Who's going to tell me the answer? Genetic constitution is genotype. Thank you, Ayush. Yes, genotype is the correct answer for genetic constitution. Now, let us look at what Mendel did. We know that he had a pea garden, he had a plant garden in which he grew a lot of pea plants. But what did he do? Okay, Mendel carried out his experiments on the garden pea plant, which is botanically called Pisum sativum. Pisum sativum, the name that you can see here, between the years 1856 to 1865. So, in fact, he grew so many of them. Like I said, he considered them as their own children. He was that fond of pea plants. Okay, so one quick question that I have to you here. Yes, Alfred, I have a question for you here. Why is it that Mendel chose pea plants? Why is it that he chose a plant in the first case? Why is it that he did not study any animal? Any answers? Well, the answer is simply that if Mendel had chosen an animal or a human being, yes, some of your answers are correct because he had a garden of it. Okay, your answers are all interesting and correct. But why he did not study human beings or animals is basically for the simple reason that Mendel himself was a human being, of course. So if he had to study many generations of another human being, his one single lifetime wouldn't have been enough to observe many generations. It's as simple as that. Plants generally have a much shorter lifespan, and pea plant specifically has a very short lifespan, and it has a lot of other qualities such as what you see here. It of course has a short life cycle. It can be grown throughout the year, which is important for a botanist or a botany scientist, right? Because availability of his or her material has to be there throughout the year, right? Now, it can be easily self and cross-pollinated. Like I said, what is self-pollination? What is cross-pollination? Remember that. It exhibits 7 contrasting pairs of characters. Very important. We will be looking at what those 7 contrasting pairs are very soon. And very importantly, one single cross results in a large number of offspring, which provides Mendel with enough sample size to come up with accurate results. Just based on the study of one single generation or one single pea plant, Mendel could certainly not have come or arrived at any of the conclusions that he did. Do you know any idea? Do you have any idea about how many pea plants he had grown by the end of his experiments? Well, I've heard that he had grown about 29,000 or 30,000 pea plants by the time he had finished his experiments on inheritance. Okay, that is quite a lot. So you can imagine his dedication. This is what a pea plant looks like. Pea, yes, of course, as a vegetable, all of you have seen it. Now, so these were the reasons Mendel chose the pea plant Pisum sativum for his inheritance experiments. This is very important to remember, children. I have seen question papers in which this is asked as a question. Why was the garden pea plant chosen by Mendel? So this is the answer that you will have to list out. Okay, so going on, I told you that we have 7 different contrasting pairs of characters, right? That is what we have on the next slide here. Contrasting pairs, I mean, one is a dominant trait and one is a recessive trait. What is dominant and what is recessive? We will look at it very soon. These were the 7 different characters. Flower color, flowers of pea plant, Mendel noticed, could either be purple or white in color. He noticed that plant height could be either tall or dwarf or short. Seed color, seeds of the pea plant could either be yellow or green. Seed shape could either be round or wrinkled. The pod color, the seed pod, right, in which the seeds are arranged. That is what I mean by pod. The pod color could either be green or yellow. Pod shape could either be inflated or full, or be constricted or flat, and flower position could be either axial or terminal. Okay, so these were the 7 different characters that Mendel considered, along with their respective traits or the respective contrasting pairs of traits or versions of the character that he studied. Why is it called dominant and recessive? We will look at it in the next few slides. Okay, so I hope that much is clear. Let us go to the next one. What exactly did Mendel do? Let's look at that step by step. What did he do first? Mendel raised pure lines with selected contrasting traits. Now, in our class, for instance, to make it easier to understand, we will consider just one character that Mendel studied, which was height. Of course, he did study all of these 7 characters. We just need to understand the basics using one character. Let's consider plant height to be that character. Okay, so why he raised pure lines with the selected contrasting traits? What do I mean by pure lines? Okay, well, that simply means that, well, pure lines simply means that say for instance, a tall pure line plant or a tall purebred plant is one that has been tall for many generations. Its ancestors and its ancestors, the parents' ancestors, grandparents' ancestors, all of them have been producing only tall plants for many generations. That is a purebred tall plant. Okay, and a purebred dwarf plant or a purebred short plant is one that has been producing only short plants or dwarf plants for many, many generations. So, pure line tall plant and a pure line dwarf plant, he cross-pollinated them. He cross-pollinated flowers with those having the respective contrasting traits to form what we call the first filial generation or the F1 generation. How did he manage to do this? This is how Mendel did the cross-pollination, or any botanist would do, to prevent self-fertilization, because like I said, pea plants can either be self-fertilized or cross-pollinated. He wanted to cross-pollinate them because he wanted to cross between traits that he chose. He wanted to cross one tall plant with a dwarf plant. He did not want to allow them to self-pollinate. Okay? So, in order to prevent self-fertilization, Mendel cut off the immature stamens, which is the male reproductive part of a plant, and he then dusted the carpel, which is the female reproductive structure, with pollen from another plant's mature stamens. Okay, so the carpels of one plant of the female parent that he chose as the female parent was dusted with pollen from another plant which Mendel chose as the male parent. And thirdly, the carpel matured into a pod containing seeds, and once planted, these seeds grew into offspring plants. Yes, emasculation is basically the removal of the male reproductive part or the anthers. That is exactly what Mendel did here. Okay, so for now, just understand that Mendel selected two pure lines, one tall and one dwarf. He cross-pollinated them, and the first generation that he got is called the first filial generation. In short, the F1 generation. Okay, what did he do next? The F1 plants were self-fertilized, and the resulting second filial generation or F2 generation were observed. The second filial generation, what did he do with this? All tall. Now, remember children, one very important thing to understand here is that here it's written all tall. What do I mean by that? It means that it was not just one plant that we had obtained as the result, or Mendel had obtained as a result. It was many plants. We are only representing it in ratios when it's a cross. Okay, remember that, which is why I've written here all tall. Okay, how many ever numbers of plants he got, they were all tall in the first generation. What he did next was he made it a point to self-fertilize them. Okay, which means he basically picked up two plants in the same generation, sister plants or brother plants, whatever they were, he made them pollinate with each other, or one same plant pollinating with itself. Okay, that is what he did. And the second generation he got was called the second phylyl generation, or F2 generation. Look at what he got there. The first generation, of course, because it's a tall and short parent, all tall, okay? The child, of course, we don't say child or children in the case of plants, but just to make it easier to understand, I may be using the terms child, grandparents, and so on, okay? So, you have seen that the children here are all tall, but in the next generation, again, when two tall plants were crossed, he got tall and dwarf plants in the ratio of 3:1. 3 tall plants to 1 dwarf plant. How come that happened? Because the parents of this generation were all tall. How come a trait from the grandparent suddenly came after skipping one generation? This was one question that Mendel was really bothered about. He tried multiple experiments, number of experiments, the same thing he repeated over and over again, and finally he saw that the results were pretty much the same. And eventually he came to all of these hypotheses. He formulated or he listed out these as his observations. In an organism, there is a pair of factors, okay? There is a pair of factors that controls the appearance of a given characteristic. Mendel called them factors. Back in his times, nobody knew about genes, nobody knew about alleles, so Mendel called them factors. Okay, makes sense, right? Factors which determine something, that's how we say. So factors, he understood that there is a pair of factors or two factors controlling one single trait, one single character. The organism inherits these factors from its parents, one from each, and a factor is transmitted from generation to generation as a discrete, unchanging unit. From parents to offspring, these factors are inherited. That's basically what the second point says. Okay? CBSE, yes, doesn't matter. This will be useful for you. The next point is when the gametes are formed, remember meiosis. Again, children, very important to understand meiosis. When the gametes are formed, the factors separate and are distributed as units to each gamete, which means that a gamete, because it is a haploid cell, it will have only one of this pair of factors. Initially, it is a pair of factors, but when gametes are formed, only one of these factors will go into each gamete. Okay? If an organism, the last point, has two unlike factors, which we call alleles, for a characteristic, one may be expressed to the total exclusion of the other. What do I mean by that? You saw here that we have two alleles. One is for tallness, one is for dwarfness or shortness. Here, only one of it expressed itself. That allele is called the dominant allele. The other allele that did not get expressed, in this case, it's the allele for dwarfness or shortness. It is called the recessive allele. Okay? So dominance and recessiveness, that is what we have mentioned here. Now, let us try and understand what this cross would look like. Tall and short, we have said that, but if we had to use alleles and write it, what would it be like? This is how you would write it. Let's represent the parent generation as the P generation. Okay? Tall plant and the dwarf plant. Tall, because it is two factors and it is pure line, the two alleles will be the same. Pure line means it is homozygous, and dwarf plant will be homozygous for dwarfness. And like I said in the previous slides, during gamete formation, G stands for gametes, only one of these would enter into each gamete. Gametes are generally represented with a circle around it, okay? These are the different gametes. And the first generation or the filial generation would have combinations of these. Now in this case, whatever combination you choose, you will get all of them to have capital T, small t as the genotype. F1 had capital T, small t as its genotype, and Mendel had observed that all of them are tall. Although the dwarf allele was here, it did not express itself, right? It is the recessive allele, which is why only dominance is expressed here. Now, what did he do? He self-fertilized this. What happens there? It is now TT and the TT, which are now the parents. Okay, what do we get from that? Let us separate out these into their gametes. It will be a capital T, small t, capital T and the small t. And different combinations of these, what would you get from those? Who is going to tell me the answer for this? Yes. What are the combinations here? Capital T and capital T. This will be a capital T and a small t. Again a capital T and a small t. And here it's small t and a small t. Which means genotypically I have one capital T, capital T. This is the F2 generation, okay? In the F2 generation, genotypically I have a capital T, capital T. I have two capital T, small t. I have one small t, small t. 1 is to 2 is to 1 is my genotypic ratio, whereas 3 is to 1 was my phenotypic ratio. What do I mean by that? Both of these genotypes will phenotypically be tall, whereas this will phenotypically be short. Because of which 2 plus 1, 3 tall and 1 short or 1 dwarf. Okay, I hope this is very, very clear to you children. Just for your reference, I have also put it here in this form. Later, if you are looking at the session and you want to understand it in a clearer manner, this is how you will have to write. Even for your exam, you will have to write it very neatly without any confusion. Parent, gametes, F1, gametes, F2. Okay, and you can also represent it in a form called the Punnett square, which is this. In this case, basically, you are just separating out the gametes. Sorry. You're just separating out the gametes here of one parent and the other parent. We are talking about the second cross or the selfing of F1, and the results, which are F2, are what you see here. 1 capital T, capital T, 2 capital T, small t, and 1 small t, small t. This is what we call as the genotypic ratio and the phenotypic ratio. Very, very, very important, children. This is not a compulsory method, but the Punnett square, we call it the Punnett square because of the person who discovered it. Okay, Punnett square, it's a very organized way. It gives you, it can give you the combinations or the genotypes without any confusion. That is why people generally prefer this. For a monohybrid cross, you can even do the calculations without a Punnett square. Okay, so that is about it. Now, this is a homework question for all of you. Okay, note down this. If the character that I'm considering is seed color, and I'm telling you that yellow seed or capital Y is the allele that's dominant to the green seed or small y, what would the F1 look like phenotypically and genotypically? You have to tell me all of that. And F2, what will be the F2 genotypic and phenotypic ratio? How many yellow, how many green, how many genotypic combinations. You have to tell me all of those, okay? I'm giving you this as homework. You have to fill up this Punnett square by yourself. If you would like to do it, you can even take a snapshot of it and post it in the comment section when you give me your answer, okay? Now, quiz time again before we end the session. Very quick, children. The genotypic ratio in F2 generation of a typical monohybrid cross would be, I'm asking you about genotypic ratio. Yes? What is the answer going to be? 3:1? 1:2:1, AK motivation, yes. Genotypic not possible. Why not, Ayush? A typical monohybrid cross. You don't have to memorize it, children. If you understand the logic, you can just sit and work out this ratio even when you are in the exam hall. Understand what it is, work it out, and you will know. Genotypic ratio is 1:2:1. 3:1 is the phenotypic ratio. The type of allele which represents itself even in a heterozygous condition, is it dominant or recessive? Yes, very quick. Dominant, 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 dominant. Yes, all of you have given me the right answer. It is certainly dominant. Mendel used the name factors for genes, chromosomes, alleles, or all of these. Slightly tricky, I guess. It is a multi-select question. More than one answer is correct, I'm telling you. Yes, D is the answer. No, D is not the answer. It's in fact A and C. Mendel did not differentiate between genes and alleles. He called both of them factors. Okay? Now, this is again another homework question for you. I don't want the answer right now, but yes, I will certainly look at who gives me the first answer. Alleles are found on sister chromatids or homologous chromosomes. Understand this concept very, very clearly. I don't want to get any random answers. If you have not understood the concept, do go back to the sessions on meiosis and have a look at that. Do understand the differences between all of these terms, children. They are absolutely important basics. And if you have not downloaded Vedantu's learning app, children, do download it right now. Join our Telegram group and stay in touch with us. And most importantly, if you liked the session, if you have enjoyed the session, do not forget to hit the like button right now before you sign off. Do share it with any of your near and dear friends if you think they would benefit from this. And very importantly, do remember to subscribe to our channel if you have not subscribed to Vedantu 9th and 10th. In the meantime, if you have any confusion, it's a slightly complex topic. I understand that, but a very important one and a fun to learn chapter. I'm telling you, children, as long as you can understand the basics in this chapter, it's very, very easy to score marks, and it is certainly one of the favorites of examiners. So you can be sure of scoring a lot of marks if this chapter is thorough in your heads. Okay? So do make it a point to attend all the remaining sessions until we finish, at least until we finish all of the basic topics in genetics. In the meantime, do connect with me on my email ID ambika.gopalakrishnan@vedantu.com or post in your comments in the comment section below. I'll see you again tomorrow in a much more interesting session. Let us continue what Mendel did in his experiments after this. This is just half of the story. We have a lot more waiting for you in tomorrow's session. Until then, happy studying and bye-bye. Good night, children. Thank you very much for being with me today.